Today we will discuss about this RMC-6263 Pharmacodynamics and Pharmacokinetics. In the previous video we have understood that this RMC-6263 can act on multiple RAS mutations to inhibit the downstream signaling pathways caused by those mutations. And basically we have seen the results for KRAS mutations and NRAS mutations. In these results, the results that are presented in this figure is focused on KRAS mutations. So what they did is they used the different pancreatic cancer cell lines and make a xenograft model into the mice. And the base results that are presented here is started from the KPN2 cell lines. So in the KPN2 has these mutations of the KRAS, that is G12W or T, G12V, W or T. Yeah, this is what is shown here. And so what it is, they first make this KPN2 xenograft model of mice and then add this drug, that is RMC-6263, at different dosage 3, 10, 25, and then check the levels of the RMC-6263 in the blood and also in the tumors. Basically they are checking the pharmacokinetics. Pharmacokinetics means what body does to the drug, that is how long the drug can survive or survive within the body. So here we can see that in the blood the levels of this RMC-6263 goes down in less than 12 hours, whereas for the tumors it continues to remain there even after 48 hours. That is EC90 for the of tumors is 809 nanomolar per liter. Whereas for the blood it is around 90 nanomolar per liter. For blood they are checking EC50. Afterwards they check this pharmacodynamics, that is how this drug affect into the body. So basically they are checking this DUS-P6 mRNA expression levels, as once the RAS signaling pathways are been inhibited, or once the RAS has been disrupted, that is RMC-6263 is will disrupt the GTP bound RAS state, so it will disrupt the downstream signaling pathways. So what the downstream signaling pathways is mainly regulated by, or mainly reported for the downstream signaling pathways is DUS-P6 mRNA. So they check this DUS-P6 mRNA levels in this different dose dependent injections of this RMC-6263, and they are able to see that even in this only in the single dose dependent, only in the single dose of this RMC-6263 they are able to see reductions in this DUS-P6 mRNA level more than 90%. And when they did it for the regularly for 7 days, they are able to see sustained decrease of this DUS-P6 level. This indicates this pharmacodynamics. And after that they compared this pERK levels also. This pERK is one of the proteins that formed in downstream signaling pathways of this RAS. So pERK levels, positive levels also goes down even in the single dose and also in the multiple dose. And these things histology images are shown in this panel C bottom side, and the quantifications are shown on this top side of this panel C. Afterwards they did the final actual studies, that is whether they are able to suppress the tumors in different pancreatic cancer cell line based xenograft model. So for the KPN2 they are able to see sustained tumor suppressions compared to the control. And similarly for this NCI-H441 model, and also for this HPAC model and NCI-H358 model. And one thing that we need to know that all these models have different kind of mutations. That is, NCI-H358 model has G12C mutations, HPAC has G12D mutations, KRAS has this KPN2 has G12V or WT mutations, and this one also has the same as KPN2, G12V or WT mutations. So this in all together indicates the role or validate the rules of this RMC-6263, that it can act on multiple mutated RAS, whereas the other drugs that are formed target on single mutations of this RAS. This could this had right now from the results it seems like it has a potential to have a better efficacy compared to the previously single mutated targeted drugs. We will discuss further results in our upcoming videos. That's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.